previous lectures we have discussed about the methods for detection and quantification of radioactivity. We have discussed three methods, one based on ionization of gases where GM counters are utilized, second one on the based on the excitation of solutions and solids where we have discussed about solid and liquid scintillation counters, and third one that is based upon the exposure to photographic emulsion, that is autoradiography. All three methods have particular applications and they could be utilized as per the requirement of in particular application. Now, in this lecture we are going to extend our discussion on radioactivity, and we are going to discuss about different applications of radioisotope technique and the safety aspects involved here. Now, before we go into that, let us first discuss this one more technique which is very important, and it is a very sensitive and versatile technique, that is radioimmunoassay. And this technique is quite widely used in both research and diagnostics. Now, this technique as it utilizes radioisotopes, this will certainly require to follow certain precautions, as well as you have to take license to use the radioisotopes. Another technique which has kind of replaced radioimmunoassay is ELISA, which we will be discussing in some other section. So, radioimmunoassay is a very sensitive in vitro assay used to measure concentration of antigens by the use of antibodies. Now, this could be like, say, you would like to determine hormone levels in the blood or certain other analyte in the blood, then if you have the suitable antibodies for that, you can use it for assaying that particular substance. Now, this technique has revolutionized research and clinical practices in many areas. For example, blood banking, in diagnosis of allergies, say, in field of endocrinology and so on. So, it has really revolutionized a lot of different areas. It is a very simple technique, very simple methodology is followed here to perform the assay. So, what is done is, I will show you the figure also, but just we will discuss little bit before that. What is done is a known quantity of antigen is made radioactive by labeling it with mostly gamma radioactive isotope of iodine, which is attached to tyrosine. Now, the radio labeled antigen is mixed with known amount of first antibody for that antigen, so that they bind to one another. So, what you have is a radio labeled bound antigen to first antibody. Then a sample of serum or patient from a particular patient containing unknown quantity of the same antigen will be taken and it will be added, and this is unlabeled antigen. So, we can call it cold antigen or unlabeled antigen. Now, what will happen is the unlabeled antigen will compete with the radio labeled, or we call it hot antigen, for antibody binding site, and this will certainly lead to at increasing concentration of unlabeled antigen. What will happen is that increasing amount of radioactive antigen will be displaced from the antibody molecules, and thereby reducing the ratio of antibody bound radio labeled antigen to free radio labeled antigen. So, what you will are going to see in the solution that you have lot of free radio labeled antigen, because it has been replaced by the cold antigen. Now, antibody bound antigen is separated from the free antigen in the supernatant, and radioactivity of each is measured using a gamma counter. So, you can like first thing to be done is you have to have known standards, and a binding curve can be generated, which allows the antigen amount in the patient's serum to be derived, as we do it for many other assays. So, this is a very simple methodology which is utilized in radioimmunoassay. Now, to explain that through a figure, if you see on your screen, if you can see here, there is a antibody here and there is a red colored antigen bound to that. That is a radio labeled antigen. Now, after radio labeled antigen you have bound it, then that is the first, we call it first antibody, and this is hot antigen in terms of it is radio labeled antigen. Now, what you do is you add unlabeled or cold antigen, which is we have negative here shown. Now, this one, as the concentration is higher or increasing, they will compete for the same site actually, because they are the same antigen, and so some of the antigens, that is radio labeled antigen, will be replaced in here, and then what will happen, that the free radio labeled antigen then could be, so radioactivity of supernatant will be equivalent to free antigen, and radioactivity of precipitate will be bound antigen. So, you can measure the radioactivity in free of free antigen, and then ratio can be taken, and finally, and this one here which is shown here is the second antibody which binds to the first antibody. So, this way you can calculate the ratio, and a standard curve can be drawn, and through which you can through the standard curve you can determine the concentration of unknown antigen. So, that could be done without any problem. So, that is the very simple method of radio immunoassay. Now, this is, like I said, it is very sensitive technique, quite used quite a lot in research as well as clinical practices, but ELISA has kind of replaced it. Now, when you have done radio immunoassay, the one thing which has to be done is separating the bound antigen from free antigen, and there are several ways to do that. You can precipitate the antigen antibody complex by adding second antibody directed against the first, like rabbit antibody can be used by adding an anti-rabbit antiserum. Then antigen specific antibody can be coupled to particles like Sephardex, and by centrifugation they can be separated. Radio immunoassay is widely used because of its great sensitivity, but will certainly have certain disadvantages. For example, both iodine 125 or 131 emit radiation that require spatial counting equipment. Now, many times body can concentrate iodine atoms which can be radioactive or not in thyroid gland, where they are incorporated in in thyroxine actually. But beside these disadvantages, despite these disadvantages, there are various applications which have been like for say hormone label in the blood. It could be very useful for the presence of say hepatitis B surface antigen in donated blood, that could be in blood banking, it could be analyzed. Anti-DNA antibodies could be utilized in systemic lupus, erythematosus like SLE. So, a lot of different applications could be there for radio immunoassay. So, this is like a very sensitive and specific technique and which could be used with radioisotopes. All right. So, now we will move on to the applications of radioisotopes. There are many applications of radioisotope technique, and radioisotopes are used in many different fields which include biological sciences as well as other fields, which even include like humanities and arts, like we will see how that is applied in there. So, let us start with the radioisotopes in biological sciences. What are different applications of these in biological sciences? Now, one is biochemical analysis. Say in biochemical analysis, you can detect presence or absence of some radioactive material when they exist in low concentration. So, radioisotopes can be used to label molecules of biological samples in vitro, that is out of the body, and thus could be used to determine constituents of blood, serum, urine, hormones, antigens and many drugs by use of associated radioisotopes. So, you can have like radio immunoassay could be utilized to determine these, like we have discussed earlier. So, biochemical analysis requires or can use different kinds of radioisotopes. Then lot of different aspects of metabolism, where research is going on in metabolism, radioisotopes could be utilized. For example, radioisotopes are frequently used for tracing metabolic pathways. It involves adding a radioactive substrate, now then extracting and separating products from samples taken at various times. Uh, One example could be to predict the fate of individual carbon atom, say 14C in acetate through the TCA or Krebs cycle. Now, this method have been developed to isolate the intermediates and know the distribution of carbon within each intermediate. Uh, Another example could be evaluating pathways of, say, glucose catabolism. Now, to know the relative contribution of glycolysis followed by Krebs cycle together with the pentose phosphate pathway, both pathways involve complete oxidation of glucose to CO2, but the origin of the CO2 in terms of the 6 carbon atom of glucose is different. So, that could be ascertained. So, lot of like in different investigative aspects of metabolism, this could be utilized. Then you can determine metabolic turnover number or how many times this takes place actually. So, radioisotopes provide a convenient method of ascertaining turnover times for particular compounds. Example, turnover of proteins can be followed by injecting a radioactive amino acid of choice, and the radioactivity in the organ or tissue of interest is determined at different time intervals. Then you can study absorption, accumulation and translocation of particular analyte. So, radioisotopes are frequently used to study the mechanism and rates of absorption, accumulation and translocation of inorganic and organic compounds in both plants and animals. So, this is another important application of radioisotope. In research in molecular biology techniques, radioisotopes are used quite a lot. The use of radioisotopes has been critical, I would say, in advancement of molecular biology, leading to genetic manipulation. The radioisotopes have been used in many procedures, including DNA and RNA sequencing, DNA replication, transcription studies, synthesis of cDNA, recombinant DNA technology and several other studies. So, in molecular biology this has really helped in understanding lot of different mechanisms and procedures. In ecological studies, radioisotopes could be used. For example, radio tracers can be used for studying migratory pattern and behavior pattern of many animals. Then examination of food chains, where the primary producers can be made radioactive and the path of the radioactivity followed throughout the resulting food chain. So, this is another important application of radioisotopes. Radioisotopes could be utilized for sterilization of food and equipment. Now, very strong gamma emitters are widely used for sterilization of pre-packed foods such as milk and meats by irradiation. Normally, it could be cobalt 60 or 137 cesium is used for irradiation. Care needs to be taken to ensure that food product is not affected, and so the doses are reduced or has to be standardized. Now, food spoilage can be reduced to a great extent by this, and it makes it more safer and fresh for a longer time. Then also these things could be these isotopes could be radioisotopes could be utilized for sterilization of plastic disposable equipment such as petri dishes and syringes, and in sterilization of drugs that are administered by injection. So, this is like quite good or quite important in sterilization of food and equipment. Another important application could be generating mutagens. Radioisotopes may cause mutations, particularly in microorganisms, and in various microbiological studies mutants are desired or desirable, especially in industrial microbiology. So, radioisotopes could be used for mutagenesis and for development of a new strain of a microorganism that could give higher yields or a desired product which could be a microbial product. Radioisotopes are widely used in a lot of analytical applications. For example, in enzyme and ligand binding studies. to study mechanism of enzyme action, ligand binding studies could be done through radio tracer method. The method is more expensive, but provides a very high degree of sensitivity. Then isotope dilution analysis could be done, like many compounds in living organisms which cannot be assayed because of presence of low amounts, and in mixtures of several compounds can be assayed by this particular method. Only thing is that you should have a radioactive form of compound, it should be available. Now, in isotope dilution analysis, what it provides is convenient and accurate method to solve this problem and avoid necessity of quantitative isolation. The technique is widely used, say for studies on trace elements. For example, to measure amount of say iron in a protein preparation, 59 Fe is mixed with the protein and a sample of iron is subsequently isolated and assayed for total iron and radioactivity is determined. So, this way you can determine very small amounts of different compounds. Then another very important and you must have heard about this is radio dating. You must have heard about that, okay, one fossil has been found which is so like a million years old or thousands of years old. So, radio dating or dating is the method of determining the age of an object, which could be usually rocks, it could be fossils, sediments, etc. On the basis of now this is done on the basis of concentration of a particular radioisotope present in the sample. Here it is assumed that radioisotopes begin to decay at the time of fossilization or deposition. Now, by determining the amount of radioisotope remaining or amount of decay products and from the knowledge of half life of radioisotopes, it is possible to date the sample. So, radiocarbon dating is one such type of radio dating method. Now, for say longer periods, uranium 238 or uranium 235 could be used, which has half life of around 4.5 billion years and decays to lead 206, as we have discussed in earlier section. So, if this is present in a rock and by how many half lives it has gone through, you can determine the age of the rock. For short term dating, 14C is widely used. For long term dating, uranium could be used actually. So, radio dating is a very important part of radioisotope technique. There could be there are a lot of applications of radioisotopes in agriculture field also. For producing high yielding crop seeds and speed up the process of developing superior agricultural products, radioisotopes could be used. Now, radiation pellets are used in grain elevators to kill insects and rodents. Insect control is very important application for pest populations are drastically reduced and in some cases eliminated by exposing the male insects to sterilization. So, they will have no offsprings and thus will be reducing in population. Tracer technique is used to study the rate and direction of movement of an element in a plant radioisotopes in plants. Radioisotopes are used for determining the function of fertilizer in different plants and also for reducing the consumption of fertilizers. So, you can see that radioisotopes also have application in agricultural field. Now, there are very like radioisotopes are widely used in the field of medicine. There are whole lot of applications in the field of medicine, right from diagnostics to detection to cure of diseases. One of the main field which is nuclear medicine is a very prominent field in the medical sciences. Now, nuclear medicine is a branch of medicine that uses radiation to provide information about the functioning of a person's specific organs or to treat a disease. Now, this information can be utilized by physician to make a quick and accurate diagnosis of patient's illness. For example, thyroid, bones, heart, liver and many other organs can be easily damaged and disorders in their function can thus be revealed through this technique. Radiations can be used to treat diseased organs or tumors as well. Nuclear medicine was developed in 1950s by the physicians with an endocrine emphasis, initially using I-131 to diagnose and then treat thyroid diseases. In recent years, a lot of advancement has taken, like for example, tomography like CT oblique PET, PET or positron emission tomography procedures have been established. So, a lot of advancement is taken in this place. Now, this radioisotope technique could be used for diagnosis, nuclear imaging and other applications. Like for example, radioactive tracers which emit radiations from within the body could be used. Now, tracers should have short half lives to minimize the radiation dose to the patient and can be given by injection, inhalation or inhalation orally or orally. So, they can be given by either injection, inhalation or they can be given orally. Nuclear imaging is another field in here to evaluate, like for example, brain functions or other organs. Organic radiochemicals are utilized for this purpose, and gamma camera like devices like gamma camera can be utilized to detect radiation emitted from the organs and can view organs from many different angles actually. Images can be enhanced by computer and can be viewed by physicians to detect blockages or other dysfunctional activity. It could be utilized to evaluate heart diseases, like for example, a radioisotope could be injected into the patient's bloodstream while he or she is exercising, like 32 people could be injected, say on a he is working on a treadmill, and the radioisotope travels towards the heart and allows the doctors to follow it on the screen. So, while looking at image, doctors can check for reduced blood flow through arteries, a signal of heart disease. Positron emission tomography, like we were talking about, is a very precise technique, and it could be utilized for different applications like in oncology, in cardiac and brain imaging. A combination of PET with CT scan or computer tomography scans can be very useful and could give a better detection or diagnosis. Then there is a field of radiotherapy. So, radiotherapy could be utilized to cure many diseases. Like say for example, rapidly dividing cells are sensitive to damage by radiation. Now, cancerous growth can be controlled or eliminated by irradiating the areas containing the growth precisely. It could be external irradiation where it is given externally, or it could be internal radiotherapy where you administer or plant a small radiation source in the target area. So, this could be lot of different kinds of methods could be utilized in here. There is boron neutron capture therapy where boron 10, which concentrates in malignant brain tumors, could be utilized. Patients irradiated with thermal neutrons which are strongly absorbed by the boron and producing high energy alpha particles which can kill cancers. So, there are a lot of different ways you can one can or the medical people can utilize this radioisotopes. Radioisotopes can be utilized in another field, that is pharmacological studies. For development of new drugs, whether it has a desirable effect for clinical applications, or say you want to locate the site of accumulation, you want to know the rate of accumulation or rate of metabolism, and metabolic products can be determined using radioisotopes. In each of these studies, radio tracers will be utilized. Say you can have autoradiography on whole section of an experimental animal and yield information on site and rate of accumulation. So, in pharmacological science studies, say accumulation of drugs could be seen or other analytes could be seen. So, diagnostics radiopharmaceuticals is used to examine blood flow to brain, functioning of the liver, lungs, heart or kidney, to assess the bone growth, to predict the effect of surgery and a lot of other things. A number of chemicals could be identified which are absorbed to specific organs, like for example, thyroid takes up iodine, brain consumes glucose. So, likewise, which chemicals or which substances accumulate where, it could be found out. So, with this knowledge, radiopharmacists are able to attach various radioisotopes to biologically active substances, which could be used for different applications. Radioactive form of these substances, after entering the body, they are incorporated into biological processes and they will be excreted in the usual way. So, there are a lot of examples of these isotopes. Now, like this like you can use gamma ray emitting isotopes, or you can use beta, like we have used 32P or iodine 131. These could be used for a lot of different applications. There is one called urea breath test. It is used to detect the presence of the bacteria Helicobacter pylori in the stomach, and this bacteria causes inflammation, ulcers and atrophy of the stomach. So, patient given with certain amount of urea, they will contain carbon, that is 14C. Now, if Helicobacter is there in stomach, urea will be broken down to carbon dioxide and it is released and exhaled by the patient, which could be collected in a balloon and then detected for the presence of the bacteria. Therapeutic radiopharmaceuticals is another field where radioisotopes could be used, like for example, to destroy or malfunctioning cells using radiation. Radioisotopes generating the radiation can be localized in the required organ through a radioactive element following its usual biological path. For example, I-131 we have seen could be utilized for treatment of cancer of thyroid, and there are other examples, like for example, boron 11 for metabolism and brain tumor treatment. So, these were applications for in the field of medical and pharmaceutical science. There are certainly there are other applications, like for example, radioisotopes could be utilized in industrial field also. Like for example, many types of thickness gauges exploit the fact that rays are attenuated when they pass through material. So, by measuring the number of rays, the thickness can be determined, and this process have several industrial applications. For example, in automobile industry, to test steel quality in the manufacture of cars and obtain the proper thickness of tin and aluminum. In aircraft industry, to check for faults in jet engines. Construction industry, like for example, to gauge the density of road surfaces and substances, and pipeline companies to test the strength of welds and other. So, their radioisotopes can very well be used in industrial applications also. There is another industrial application could be cable manufacturers, they to check cables for cracks and other things. So, these are like a few applications of in industry. Now, not only in industry, like I said, they could be used in art and humanity also. Neutron activation analysis is useful in identifying the chemical elements present in say coins, pottery and other artifacts from the past. A tiny unnoticeable fleck of paint from an art treasure or a microscopic grain of pottery is sufficient to reveal its chemical makeup, and this could be utilized say work of famous painters that can be fingerprinted and could be detected. You can detect the work of forgers also. So, radioisotopes, if you as we have gone through, has applications in many areas, like from biological sciences or biotechnology to agricultural sciences, to medical sciences, in pharmaceutical industry, in normal other industries, and even in art and humanity. So, radioisotopes have wide applications, and it is like it is being utilized for many different being utilized in many different areas of science and otherwise. Now, although it has lot of applications, but radioisotopes or radioactivity is a dangerous thing. It is not safe unless it is properly utilized. So, there are a lot of safety aspects are related to this. So, when you have to utilize it, you have to take permissions, due care and precautions has to be taken, and in this section we are going to discuss those safety aspects involved with the use of radioactivity. Now, radioactivity is a natural phenomenon, and natural sources of radiation are features of the environment. They are present all over in different forms actually. Now, radiation and radioactive substances have many beneficial applications like we have seen, ranging from power generation through nuclear plants to use in biological sciences, in field of medicine, in field of industry, agriculture and likewise. Now, the radiation risk for to workers, to public and to the environment that may arise from these applications has to be assessed, and if necessary has to be controlled. So, that is a very important part of radioisotope technique. So, therefore, some of the activities mentioned like which I am mentioning here are subject to standards of safety. For example, medicinal use of radiation, one has to take care that how much doses has to be given, whether they are safe or not. Then operation of nuclear installation, where they are localized, like are they close to community, what are the safety precautions being taken to operate them. And then production and transport and use of radioactive material. Then very important part is management of radioactive waste, which is generated after its use. So, it has to be properly disposed, so that it will not cause any harmful effects. Now, greatest disadvantage of using radioisotope is their toxicity, and they are toxic because they produce ionizing radiations. So, when absorbed, radiation causes ionization and free radicals that will interact with the cells and different parts of the cells, macromolecules, and which could cause mutation in DNA by affecting or altering the DNA, and could hydrolyze proteins. Now, these can damage any molecule by ionization, and maybe damage to DNA for certain extent could be repaired, but not all the time. Many times the DNA damage is not repaired, and it could lead to mutations, and which could be deleterious also. Like for example, mutation can lead to the formation of tumor, and this has happened in many cases. For one example, when we are utilizing these radioisotopes in labs, in biotechnology labs, there are certain, or any biological lab, there are certain good lab practices for safe use of radioisotopes, and I would like to discuss some of them in here. Now, good lab practices is a must for anybody who is going to use these radioisotopes in any part or anywhere or for any application. The first part is one has to become fully conversant with radiation protection by reading lot of literature, like reading regulations for the safe handling and use of radioisotopes. There are a lot of publications like code of practices to use radioisotopes and other publications which are published by competent authorities. So, those needs to be read thoroughly and understood thoroughly to before going on to use the radioisotopes for your research or other works. Second thing to be done is one needs to obtain a license from the Department of Energy or Department of Environment. Like for example, in India you have to obtain a license from Bhabha Atomic Research Center or BARC and other agencies. And second part is that before you are utilizing radioisotope technique, you need to get the proper permissions, you need to obtain license, and they will be monitoring your area of research, where you are going to work in all different aspects, so that a safe utilization of radioisotopes could be performed. Then when you are using it, after you have done these first two things, then there are certain norms to be followed, like proper training has to be taken, and when you are utilizing radioisotopes in the lab, first thing is protective clothing, that you have to protect yourself. In protective clothing, you have to wear gloves which could be protects so that you do not come in direct contact through your hands, there could be double gloves could be worn, there should be full length lab coat should be used, and closed toed shoes like completely closed shoes has to be utilized, and then safety glasses for protection of your eyes. So, you have to protect yourself, your sensitive areas like skin, eyes and other you have to protect them through protective clothing. Second thing is mouth pipetting needs to be avoided completely. You will not mouth pipette a radioisotope solution. So, what you have to do, you have to use automated pipettes for this purpose. There should not be any food and drinks in vicinity where you are utilizing radioisotopes or you are storing radioisotopes. Food and drinks has to be kept at a safer distance from where your area is where you are utilizing radioisotopes or where you are storing. Like for example, if you are storing at certain place, food and drinks has to be kept away from that place. Storage of radioisotopes is another important area. You will not leave radioactive material unsecured and unattended. Only when you are present there, you will be utilizing those radioisotopes, and otherwise you will lock and secure those radioisotopes in safe place. So, this is a very important part and one has to really pay attention to this. Many times it might happen that you might spill radioisotopes while using them, and so spillages are another important problem. So, all spillages like which are maybe on the floor or your working area or on your skin or clothing should be attended immediately, and due care should be taken, and it should be problem should be solved here. Spillages should be avoided, and but if it is spillages occur or you have spilled the radioisotope, you should clean it up, and proper guidances or directions needs to be followed here. All the labs which are utilizing radioisotopes has to use proper signs and labels. So, any container, equipment, room, contaminated area with radioactivity should be labeled with proper signs as radioactive. It's it should show that it is a dangerous thing, and one has to be careful not to go near it unless somebody needs to use it, and that too with protective clothing and all other precautions. I will show you the signs what are used, like some of those. Then there should be like radioisotopes should be used in safety cabinets or fume hoods. There is a term called ALARA, ALARA, which is like here ALARA, that is keeping radiation exposure to as low as reasonably achievable. That is what it means. So, one has to take all the precautions to follow all the guidelines to keep radiation as low as reasonably achievable. So, what one has to do, take all precautions to keep radiation exposure as low as possible, wear radiation monitor badges and get them checked regularly for radiation exposure. We will discuss it in a little while. Check yourself, your clothing, area of work after radioisotope work is done, and this could be done by like GM counters routinely. Even if spills have taken place, you can use GM counters to detect that, this is a very important part of safety in radioactivity use. So, these are some of the things. Now, if you can see these symbols here, this is like it shows ionizing radiation hazard symbol, which could be pasted on the areas where work is going on the lab doors of the labs which are utilizing radioisotope technique. There is another one which is radioactivity danger logo, which could be also pasted at the areas where radioisotopes are being used. So, these are two signs, this one and another one, and there are other signs also which needs to be pasted, and a proper dangerous signs has to be people has to be alerted for these where radioactivity is being used. Now, the if like little bit into what happens when a certain amount of radiation somebody is exposed to, and what are different parameters which are important in here. Now, the toxicity of radiation will be dependent on if somebody is really exposed or comes in close contacts. It will depend on, first is the amount present, that is amount of radioisotope or radioactivity present, then amount absorbed by the body and energy of the absorbed radiation and its biological effect. All these things needs to be assessed. So, therefore, a series of additional units and other ways have been devised to assess this. Radiation hazard can be measured in terms of exposure, that is a quantity expressing the amounts of ionization in air, and there are a lot of different ways which could be done. We are not going into details of that. One has to monitor and control exposure. Radiation has always been present in the environment and in our bodies. The human body cannot sense ionizing radiation, but a range of instruments that are capable of detecting very low levels of radiation from natural and main source exist, and they should be utilized. Like for example, there are dosimeters which are widely utilized. Now, dosimeters are available to measure the absolute dose over a period of time. If you know, we were talking about the badges needs to be put on your clothes or in close vicinity, where it could be the amount of exposure could be checked. So, dosimeters are available for that, and they could measure the absolute dose over a period of time. There could be ion chamber dosimeters, and they resemble like pens and can be clipped to one's clothing and must be periodically recharged, and the result should be logged. There could be film badge dosimeters, and they enclose a piece of photographic film, and this film becomes exposed as the radiation pass through it. So, they must be developed in photographic emulsion as photographic emulsion. So, the exposure can be counted and logged. So, once developed, they are discarded. There could be thermoluminescent dosimeters which contains crystals that emit visible light when heated, and they could be utilized again for logging and to record amount of exposure. There are a lot of other like Geiger counters or scintillation counters could be used for to measure these things, radiation. Now, another important part is how to limit the exposure, that is radiation protection. Now, this is also known as radiological protection, and it is the protection of people and the environment from the harmful effects of ionizing radiation, which include both particle radiation and high energy electromagnetic radiation. Now, radiation protection can be divided into one occupational radiation protection, like protection of workers, and then medical radiation protection, that is protection of patients and the radiographer. Then there is public radiation protection, like for example, protection of individual members of the public and the population as a whole. For example, if there is a nuclear plant, then that comes under public radiation protection. Radiation exposure can be managed by a combination of three factors that control the amount of or dose of radiation received from a source. For example, one is the first factor is time. The amount of radiation exposure increases and decreases with the time people spend near the source of radiation. Now, reducing the time of exposure will reduce the effective close proportionality. So, if you are near the radiation source for lesser time, you will be exposed less, proportionately less. Then distance, that is very important, that farther away people are. So, one needs to be as away from a radiation source as possible, and that will also result into the less exposure. Also, the energy of the radiation will determine whether if you are at a particular distance, so that your exposure is less. For example, we have discussed about the beta particles with different energy levels, gamma rays which are highly energetic, and alpha rays which are not so, they are energetic, but not so penetrating. Now, third factor is shielding, which is very important to protect people. Now, greater the shielding around a radiation source, the smaller is the exposure. So, biological shield refers to a mass of absorbing material placed around a reactor or other radioactive source to reduce the radiation to a level safer to humans. Now, effective for if material as a shield is related to its cross section for scattering and absorption, proportional to the total mass of material per unit area interposed along the line of sight between the radiation source and the region to be protected. There could be different radiations, like I said, alpha radiation, a thin piece of light material could be utilized, such as paper, or even the dead cells in the outer layer of the human skin are adequate for shielding, as these are not penetrating. So, but if alpha source is present inside the body, then there is no protection, because the cells could be exposed to these particles. So, if these are inhaled or ingested, then they could be dangerous. Beta particles, like I said, they could be of different energy, and they could be covered, or they could be avoided by heavy clothing, and like for example, materials of 5 millimeter of aluminum could be good enough to attenuate these rays. Gamma rays are highly penetrating, so you require thick dense shielding such as lead is necessary to protect against gamma rays, and higher the energy of the gamma rays, thicker the lead must be. Likewise, X rays, the exposure to X rays could be also shielding could be utilized, proper shielding could be utilized for that also. Now, so when you are handling the radioisotopes, the rule is to minimize the distance between yourself and the source, minimize the time of exposure, and maintain the shielding at all times. Management of radiation protection is similar in many countries, and there are, like I said, there are a lot of regulations, there are a lot of publications which one should be conversant with, and proper licensing and proper precautions need to be taken to use these radioisotopes or the radioactivity. So, this is little bit about the safety aspects which needs to be taken care of while using radioisotope technique. So, this completes our section on radioisotope technique, and we have gone through the basic concepts of radioisotope technique, where we have learnt about different types of radioactive decay, the interaction of those particles with the matter, different types of detection and quantification methods, particularly 3 methods. Then we have learnt about radioimmunoassay, very much used in research and clinical practices, and we have learnt about the various applications of the radioisotope technique, and particularly the safety aspects, which are very important for public at large, and for researchers, medical science people, to be protected from the radioisotopes while using them for a lot of different applications. In coming lectures, we are going to start a new topic, that is chromatography, and how macromolecules and other important constituents are purified. We are going to discuss that in coming lectures. Thank you.